Hello everyone. In this INR series, we are going to discuss about steps of karyotyping, which was INICT question in 2022. And from here, you will get 3 or 4 questions which had been already asked in NEET PG exam and FMG exam. So listen to this very carefully. You may get this question. In INICT question was what is the sequence of steps of karyotyping? So let's start. So how we will remember this? We can remember this by a mnemonic, and that mnemonic is Supreme Court. SC is Supreme Court. Supreme Court arrested and fixed sliding thief Mickey. Now you can see that Mickey is sliding thief here, right? So sliding thief Mickey has been arrested and fixed by Supreme Court. So this is what we have to remember. Supreme Court means S is for sample collection. We will collect the sample. C is for culture. Arrested is arrest at the metaphase. Fixed is fixative with the fixation with the fixatives. Sliding is slide making after that. And thief is a trypsin digestion and staining. And Mickey is a microscopic examination. So this is the beautiful mnemonic to remember this karyotyping. And now I'm going to tell you what are the steps we are going to see here. So as I said, what is the number one step? We will collect the sample. When a patient will come, we will take out the sample which is a venous blood. So how much? It is 5 cc of the venous blood. So sample will be connected. How much? Sample will be collected will be 5 to 10 cc of the venous blood, and then we will be mixing with heparin. So this is number one, sample collection. Second step is a culture. After collecting this, we are going to culture this blood sample, and in that culture we will add phytohemagglutinin. Remember why we are adding phytohemagglutinin? Why we are adding phytohemagglutinin? Because it will activate a T lymphocyte mitosis. Because T lymphocytes are there in the blood sample, which has to start their mitosis, and for that purpose we are having phytohemagglutinin. So that culture with phytohemagglutinin will be kept for 3 days at 37 degrees Celsius, right? So that is what second step. Second step is culture, add phytohemagglutinin and keep them at 3 days at 37 degrees Celsius. Third step is metaphase arrest, right? So remember, third step is metaphase arrest. What will be used for metaphase arrest? So remember, we are going to add alkaloid. Which alkaloid we are going to use? That is the question in exam. So please remember, colchicine alkaloid will be used for metaphase arrest, right? So please remember, as I said, this is the question which had been asked in exam. Colchicine alkaloid will be added, right? So colchicine alkaloid will be added for which phase? Metaphase arrest. Why metaphase only? Because this is the phase, remember, this is the phase where chromosomes are more clearly and distinctly visible, right? So that is what we are doing, alkaloid addition at the stage of metaphase arrest. And what we will do after that, we will wash them with the hypotonic saline, right? So now you understand, during metaphase arrest you add the alkaloid colchicine to arrest them, and after that you have to clean the sample with hypotonic saline. So add colchicine and do the hypotonic saline washing. After that, what you will do? You will do the cell fixation, right? Cell fixation will require fixative, and ideal fixative is Carnoy fluid. Remember, it is again important MCQ in NEET PG exam. Carnoy fluid is used for cell fixation of karyotyping. Remember, for karyotyping, what is the best fixative for karyotyping? Fixative of choice for karyotyping was a DNB question also, that is methanol. So what is the Carnoy fluid? Actually, Carnoy fluid is the ideal solution, which is a mixture of methanol plus glacial acetic acid. So basically, fixative of choice for karyotyping is a mixture of methanol plus glacial acetic acid, and by this method we are going to fix the cell. So now you understood, cell fixation is done by methanol and glacial acetic acid, which is called as Carnoy fluid, right? After that, we will put that sample drop by drop on this slide, so that is called as slide making. So we are dropping the sample on the slide and we are preparing the slide. This is called as a slide making. After slide making, because there are some unwanted cells also, you have to remove them. For that, you are going to digest with trypsin and then you are going to stain with Giemsa. So trypsin digestion. So remember what we are going to do: trypsin digestion and staining. So best stain for karyotyping, that is also exam question. So best staining for karyotyping is Giemsa stain, and that is why this staining is also known as G banding, or karyotyping is also known as G banding, and both are exam question. Best stain, Giemsa stain, and that is why karyotyping is called as G banding. Now you understood what is the sixth step. Sixth step is trypsin digestion with staining. Digestion with this trypsin, and then we will apply the Giemsa stain. This is why we call them as a G banding. And then we will do the microscopic examination of the metaphase spread. So that these are the total 7 steps. You have collected the sample, you have done the culture, you have done the metaphase arrest with the help of colchicine. We have fixed the cell with cornoid fluid or methanol glycolic acid. Then we have done the slide fixation. We have done the trypsin digestion, and then we had applied the best Giemsa stain, and that is why it is called as Giemsa banding. And finally, the last thing, we are going to analyze them on microscopy for metaphase spread, and this is how karyotyping band will be seen. So these are 8 steps of the karyotyping, and please remember, this is the mnemonic for steps of karyotyping. You can see this Mickey is the thief who is sliding, right? And Supreme Court is arrested and fixed sliding thief Mickey, right? So please remember these 7 steps of the karyotyping. Best wishes for your exam.